Welcome everybody. I'm Assistant Professor Dr. Reham Mosawi, specialty with Medical Microbiology in the College of Dentistry. I'm a faculty member in Basra University of Iraq country. My presentation is titled with Identification of Staphylococcus aureus by specific 16 sRNA and detection of MEK gene from the clinical samples in patients of Basra. Introduction. As we know, Staphylococcus aureus is a gram-positive bacterium and commensals microorganism that colonize about 30% of interior areas of human individuals. This bacterium play a considerable role in the causing of both nosocomial and the community-acquired infections. During the past few decades, treating infections caused by the Staphylococcus aureus has become challenging due to the emergence of the multi-drug resistance, particularly the methicillin-resistant Staphylococcus aureus MRSA. The antimicrobial resistance in MRSA strain is correlated with the question of a mobile genetic element called staphylococcal chromosomal cassette MEK, which harbors both the MEK-A or MEK-C genes, which are responsible for the production of proteins with low binding affinity for the beta-lactam antibiotics such as penicillin binding protein. Aim of study. The aim of this study is to detect and identification of specific 16 sRNA gene and MEK-A gene in MRSA isolates from the clinical samples by objectives. Number 1, isolation and identification of methicillin-resistant Staphylococcus aureus MRSA from the different clinical specimens. Number 2, molecular detection of the bacterial strains by specific 16 sRNA gene and MEK-A gene in MRSA isolates from the clinical specimens by PCR. Materials. Study design and sample collection. A total of 150 clinical specimens, including wound swabs, sputum, throat swabs, nasal swab, pus and urine, were collected from the patients suffering of the urinary tract infections, wound infections and upper respiratory tract infections. These patients attended the outpatients and inpatients clinics of Al-Sadr Teaching Hospital and Al-Shafa General Hospitals in Basra in Iraq for a 7-month period from 1 January to 30 July 2022. All samples were collected in a sterile condition with a container and transmitted to the laboratory in Department of Microbiology in the College of Medicine, University of Basrah for isolation and This slide showed work outline after septically collected of the clinical samples from the patient, transport to the microbiology laboratory in College of Medicine for the culturing, isolation of and identification of bacterial isolates as Staphylococcus aureus by using of specific and selective media, then complete identification of bacterial isolates by the biochemical test. After that, confirm detection of Staphylococcus aureus isolates by the Vitek-2 compact system and PCR detection of the distinct bands base pair corresponding to the 16 sRNA gene in the bacterial isolate. On the other side, detection of MRSA isolate with antibiotic susceptibility test against oxacillin and cefoxitin disc. Then confirm detection of MRSA by the Vitek-2 compact system and PCR for the detection of the distinct band base pair corresponding to the MEK-A gene. This slide shows primers that's used in this study for the detection of 16 sRNA and the MEK-A gene in bacterial isolates, which corresponding to 228 base pair and 310 pair for both genes. This slide shows amplification of the genes for the detection of Staphylococcus aureus isolates and MRSA isolates. A mixture of PCR, which prepared before perform of PCR technique, which include master mix, primer, forward reverse primer for both genes, and DNA template with nuclease-free water that equal to 20 This slide show PCR cycling program parameters and condition which installed for the beginning of in the machines of the PCR for the beginning of the PCR amplification of genes and the data management and statistical analysis. Collected data were fed into SPSS matrix version 24 for tabulation and the analysis. Quantitative data were examined for normal distribution using the Shapiro-Wilk and Kolmogorov-Smirnov test. It was found that the data were normally distributed. Chi-square tests were used to assess the categorical and the nominal variable. Level of significance p-value for all of used statistical tests were set to 0.0. Table 1 showed demographic features of the study population, which involve 4 age groups that involved in this study for obtain the samples, clinical samples from the patients, which include male and female, that suffering from the different types of infections. We can see in this table a high number of patients recorded in age group 30 to 39 with 26 patients, female patients that suffering from the respiratory tract. Table 2 shows distribution of the clinical samples in relation to hospitals. We can see a high percentage of Staphylococcus aureus recorded 50% from the Al-Sadr teaching hospitals, followed by the 20.83% from Al-Shafa General Hospital. This figure shows the diagnostic test for the detection of Staphylococcus aureus from clinical samples. Most important test for detection of Staphylococcus aureus than other staphylococcal species detect a positive coagulase test of detection Staphylococcus aureus and positive mannitol fermentation on mannitol salt agar with beta hemolytic of Staphylococcus. This table 3 shows the frequency of Staphylococcus aureus and the other Staphylococcus species in different clinical specimens. As we shown in this table, the high percentages which recorded as a positive Staphylococcus aureus isolate recorded 62.5% from the earbus swab, followed by the 60% from the sputum, and the other Staphylococcus species recorded high percentage with 52.38% from the nasal swab, followed by the 51.21% from the urine. The result showed statistically, the result showed a significant difference at p-value less 0.005 from the distribution of Staphylococcus aureus in the different clinical samples and compared with other. This table 4 showed species of other staphylococcal, which include Staphylococcus epidermidis and 73.21% and Staphylococcus saprophyticus and 26.78%. Table 5 showed the antibiotic susceptibility test towards 61 of Staphylococcus aureus isolates by using of the disc diffusion assay. We showed a high resistance of these isolates, of these Staphylococcus aureus isolates, recorded 100% for both antibiotic disc, oxacillin and cefoxitin, which recorded as a MRSA isolate, as shown in this figure on mannitol on molar Hinton. Table 6 showed antibiotic susceptibility test towards 61 MRSA isolate, again, which determined for the showed a resistant to the other standard different antibiotic disc used in this study. Also, these MRSA isolate showed high resistance against other antibiotic disc, which recorded 100% against ampicillin, methicillin, amoxicillin, and followed by the 60.65% against rifampin and 47.54 against azithromycin. Table 7 showed the frequency and percentages of 16S rRNA and MEK-A genes which detection in Staphylococcus aureus that isolated from the clinical specimens. Out of 61 Staphylococcus aureus isolate, confirmed detection of 16S rRNA gene in 52 with 85.24%, and out of 52 Staphylococcus aureus isolate detection 80.76% and 42 isolate as a MEK-A which have a MEK-A gene as a MRSA strain. Figure 3 showed agarose gel electrophoresis for detection of purified fragment of DNA of Staphylococcus aureus isolates with the line numbers of these Staphylococcus aureus isolates. Figure 4 showed agarose gel electrophoresis of PCR amplified for 16S rRNA gene of Staphylococcus aureus isolates. We can see the confirmed detection of 16S rRNA gene that recorded 228 base pair in these isolates, which compare with the DNA marker with base pair 1000. Figure 5 showed agarose gel electrophoresis of PCR amplified for MEK-A gene of Staphylococcus aureus isolates. This figure showed a confirmed detection of MEK-A gene in this isolate of Staphylococcus aureus, which recorded 310 base pair in these isolates when compared with. Conclusions. Number 1, PCR based 16S rRNA and MEK-A genes which used for confirmed identification of MRSA isolates in culture of Staphylococcus aureus from the clinical samples showed a high prevalence of MRSA isolates. Number 2, all bacterial isolates of Staphylococcus aureus showed resistance to oxacillin and cefoxitin and were considered as a MRSA. Number 3, high percentages of MRSA isolate showed resistant against the most of other antibiotics. Recommendations. Number 1, preferable using of the molecular techniques in the hospitals and other private medical laboratories to confirm the identification of the pathogenic microorganism and that leading to the use right antibiotics in treatment, then reducing the spread of the resistant strains as MRSA both in hospitals and community. Number 2, due to the antimicrobial resistance has emerged, spread and endured in MRSA strains, it has become imperative to develop a new and effective alternatives to traditional antibiotics like nanoparticles as antimicrobial agents for treating the infections caused by these. This figure show the references that used in this